Great. True pleasure to introduce the 8 p.m. event. This is on Multiscale Maps. We have Tarek Ashkar here. He is the Terence Khan Professor of Nephrology, Kidneys, at the IU School of Medicine, and Daria Barwinska. She's an Assistant Research Professor of Medicine, also up in Indianapolis, which is just in 1 hour away, but still we don't get to see each other often. But if we get to see each other, it's actually really fun, interesting, exciting. And it's so wonderful that you could join us here today. Thanks for coming in.
Yeah, thank you so much. We are very excited, privileged, and looking forward to, discuss a little bit of science and also just some, take some questions, have a discussion. That would be great. So.
Yeah, and keep in mind it's for general audiences, right? Like my mom, my children. I think it's not easy. Oftentimes it takes like 2 steps of translation to get from a scientific paper to a journal to kind of press item that
Yeah.
you might have in your IU press release, and then it takes one more step to get to where we are here right now. And I think it's a challenge, and thank you for taking on that challenge. And
Yeah,
if you
no,
can show any kind of behind the scenes, I would
yeah.
love that.
I mean, so what we plan to do is, I mean, I'm just gonna just get a brief introduction, introduce Daria, and Daria gonna go through a little bit discussing about the kidney and what we're doing. By the way, just for the audience, I am a kidney doctor, so I mean, this morning I was in the hospital taking care of patients with kidney disease. In the evening, we do science, we do presentations across the world. So we study the kidney also. So we take care of patients with kidney disease, but we study the kidney and we try to understand it. I think the opportunity, what we are hoping to show today, is how we kind of go from the organ down to the cell level and understand. I think the previous panelists were talking about these different cell types, and now we're trying to understand them in the context of other cells. So I think the whole idea is cells are not alone. We have what defines a cell? Is it its function? Is it the markers that it has, or is it its state? Is it an angry cell? Is it a happy cell? Is it a cell that wants to heal? Is it a cell that wants to call for backup? And I think those are important concepts that we're trying to kind of put together scientifically, but eventually translate them into things that are relevant, for example, for disease. How do we use this information to treat patients with kidney disease, to develop therapies that target these specific cell states? Another concept that we have become very interested in is who are the cells living next to? Which type of cells are they? How are they living together? Are they living in a peaceful neighborhood? Are they living in an angry neighborhood? And I think those are really important concepts to kind of try to develop that. And we could not do that without these maps that we're developing, both in health and now we're advancing to disease. With that, I think I'm going to introduce Daria, who's, as Katy mentioned, she's an assistant research professor at Indiana University. She's a senior scientist in our group, and she's really building these maps using data from different consortia to kind of come to these questions and expose this information to the world, really. And our goal is to really have this information available through the consortia that we work with, particularly, I think it was mentioned before, the HubMap consortium and also the Kidney Precision Medicine Project consortium, of course, along with other work that we're doing. So with that, Daria, please take it away.
Thank you, Dr. Ashkar, for the introduction, and thank you, Katy, for having us here. It's a pleasure to be part of this event. So with that, I will start. I hope you see my slides.
Yeah, I can see them.
Perfect. I will go into the presenter mode. All right. So I will start first with the acknowledgements. I'd like to acknowledge our Ashkar lab team and all our other nephrologists and scientists on the floor within the IU School of Medicine, as well as the Indiana Center for Biological Imaging, the O'Brien Center, and the Center for Medical Genomics. Of course, a lot of the work which went into a lot of the slides I'm going to show you today has been supported by the IU Showalter Fund. And of course, I do have to also acknowledge Kidney Precision Medicine Project Consortium, as well as the HubMap Consortium and the CNS, which is based in Bloomington. So with that, let's talk about kidneys. So kidneys are two bean-shaped organs located on each side of your spine in the lower back. And they are characterized by this very specific for the human kidney layout. So on the outside, you will see a cortex of the kidney. And then as you progress closer towards the pelvis right here, you enter the medulla space. And so nephron is one of the famous kidney functional units, filtrating units. And so within the cortex, you will see the glomeruli, which are these apparatus right here. And as they filter, and as the filtration products are passing through the nephron, they are going to pass through the proximal tubule. They're going to enter this loop of Henle right here, and then end up in the distal convoluted tubule and ultimately merge with the collecting duct. And so the glomeruli, are present in the cortex right here, whereas the longer elongated part of the nephron, the loop of Henle and the collecting ducts are going to be found in the medulla and also in the pyramids, which are these structures right here. And so as the urine starts to drain from the collecting ducts into the pelvis, that then gets passed into the ureter. So what's the kidney function? Joel Topf, a nephrologist, pointed out really well by saying that the product of the kidney as a urine is like saying that the product of a factory is pollution. Urine is a byproduct. The product is homeostasis. And I think this is really well put. So the actual function of the kidney is to remove the waste, is to balance the fluids, is to regulate the blood pressure, and to produce hormones. So how do we look at the kidney? So there are many different tools that can allow us to really dive deeper and look into specific morphology and how the structural units and how the cells are aligning in health and, of course, in the disease. And so pathologists primarily use the 2D light microscopy. And so they're able to visualize a very thin kidney section and really look at the alignment of the tubules and be able to determine any kind of pathology and diseases which are going on within that particular tissue sample. And so the method we are using in our research is what's called the 3D confocal microscope. And so this allows us to take an entire kidney biopsy. And so this is an example that you see on the screen of a needle biopsy of a kidney. And we are able to visualize the entire piece of tissue. This is actually a 3D volume, so it's a very thick stack of different planes, many planes as the microscope goes down, imaging each of them. And so we are also able to utilize a variety of antibodies to really be able to visualize different structures within that nephron that I just showed you. So we can look at the proximal tubules, which are close to glomeruli, those filtrating units. We can look at collecting ducts, for instance. And so then go really deep, zoom in right into those structures to see how they're close together, how they align with each other. And of course, make sure that they are distinctive as we visualize them using different antibodies or the markers specific for those particular segments. So this is a video that is going to allow us to dive a little bit deeper into a human kidney. So we see those circular units, those are the glomeruli. We are now looking at the phalloidin staining for F-actin. And we are looking at all the tubules highlighted. The little dots that you see in gray, those are the nuclei. So those represent every single cell in the kidney. And now we are going to enter slowly the medullary ray, kind of going down in the kidney towards the kidney pyramid. And you see how tightly all those structures are together, how those tubules are really just very close. And so using different antibodies is going to allow us to really visualize each of those tubules. You see in green now here on your left, there is a blood vessel. And different tubules can also be distinguished by the different density of those nuclei within each of the tubule. And we are coming out. This is screenshots going through each of the staining channels that we are using in our 3D model. And I think that's finished. All right. So we talked about the kidney structure, the kidney function, and of course, it's important to know that sometimes things go wrong. And chronic kidney disease in this country accounts for close to 37 million people being affected by CKD, and that amounts to 1 in every 7 individuals, which truly makes chronic kidney disease the 9th leading cause of death. I'm also wanting to stress that kidney disease has several stages of progression, and that's 5 stages. The first one being the normal kidney function with the healthy kidney. And as the impairment gets worse and worse, we progress towards stage 5, which is the kidney failure. So there are also a lot of different factors which can affect and contribute to developing of a kidney disease. The list right here on your left is not exhaustive, but some of the more important ones and the more common ones are the diabetes, obesity, uncontrolled blood pressure, infections, heart disease, different medications. And so all those can affect the well-being of the kidney and ultimately result in different types of kidney disease. And so the list on your right is also not exhaustive, but I did try to list those which are perhaps the more common ones. So those would include diabetic kidney disease, the acute kidney injury, lupus nephritis, IgA nephropathy, even kidney stones. So now the next question is, with so many potential diseases that we are trying to target and try to find cure for those and really understand what's the mechanism underlying them, what can we use to investigate the kidney, to assess the differences and the changes from the healthy state to the disease state? And so spatial technologies have been truly up and coming in the last decade or so. And within the spatial technologies, we have several different types. So we have codecs, we have IBEX, we have cyclic IF, we have spatial transcriptomics, imaging mass cytometry, imaging mass spectrometry, multiplex ion beam imaging. And so among all of them, we are going to focus in this talk on codecs. This is something which our lab primarily has a big focus on. And you would ask, why even are we involved in spatial technologies? Why is it important to us to keep utilizing them? And so when you think about the information that can come from cellular and molecular aspects kind of within the tissue, that are hidden within the tissue, and also at the same time preserving and being faithful to the layout and to the location from where that information is coming from. That's actually crucial, and that's really putting us a step forward to understanding and getting closer to understanding kidney disease. So that's really where the advantage of spatial technologies come into play. So I will focus here on codecs. The principle of it is quite straightforward. You adhere a section of a kidney onto a microscopy slide, and you flood it with different antibodies. Those antibodies stand for different markers which you want to image. And then you flood the tissue with barcodes, which are corresponding to the antibodies that you have on that tissue. And so as you do that, then you proceed to image the particular section, and then you remove those barcodes, and then you do the same process over and over. And so it's this kind of cyclical manner of adding and removing the barcodes, which allows you to visualize different antibodies, and as a result, different structures within the kidney. And those could be the structural cells. So for instance, the glomeruli, or those could be immune cells, or those could be different cell states. So a cell that's healthy or cell that's perhaps injured. So I will briefly describe another technology, which I'm also going to just briefly mention in this talk, that we also utilize, and that's Visium spatial transcriptomics. So there is several iterations right now of the spatial transcriptomics. There is Visium ST, there is Xenium ST, there is also Visium HD, which is a little bit more granular, subcellular level of resolution. But essentially, you adhere tissue onto the microscopy slide, a specialized slide. And on that slide, you'll find those dots, which are on the image on the bottom. And so within each dots, as you overlay with the tissue and then you do sequencing, you are able to then pinpoint the reads in the sequencing. Where are they exactly coming from? And you can really determine where a specific RNA read came from, which region of the tissue. So that's the principle of Visium ST. And so I'd like to also point out that Nature Methods just recently announced that spatial proteomics is the method of the year 2024. So this is exactly what we are going to talk about today. So as I'm going to walk through the slides and show you this research that we are working on, I will present to you right now the panel of all the markers which we have utilized to obtain the data, which I'm going to share. But of course, within our lab, we are continuing to optimize the number of markers and expanding the number of markers. But right here, what we have is, of course, you've seen the nephron already. And so we have all those different structures and parts, segments of the nephron. So for every single structure or segment of that nephron, we have markers. So for instance, in podocalyxin is a marker that we know would stain glomeruli. For instance, LRP2 would stain proximal tubules, and so on. We have markers for the loop of Henle, for the thick ascending limb, as well as for the distal tubule and for the collecting duct. But that's not all. We also have markers that are strictly structural. So those will be the collagen, the vimentin, the alpha SMA. And at the same time, we are also visualizing endothelium and immune cells, which are also very crucial to be able to visualize within this entire context of the kidney tissue. And last but not least, we are also looking at cell state and cell injury markers. And so here we have, for instance, Ki67, which will allow us to look at cell cycling. We have phospho-MLKL, which looks at necroptosis. We have markers which would look at autophagy, and of course, a variety of different injury markers. So with that, I will show you here, these are 3 panels of structural markers, immune markers, injury markers, exactly as we are able to visualize them using the Codex platform. And this panel actually went into a very recent manuscript, which we just deposited a few weeks ago on the bioarchives. So this is essentially how the data comes out of the microscope of Codex. And so once we visualize all of these tissues, how do we do the analysis? So once we have an image, so for instance, the example of the image on the very left, we then process those images, doing cell segmentation. And so as we identify each individual nuclei, each of the nucleus being kind of a fiduciary for a cell, we identify those nuclei, we know which marker they are associated with. So for instance, we know this nucleus is associated with a marker for collecting ducts. This one for endothelial cells, for the vasculature. And so we are then able to pull that information together and then sort them into specific clusters, clusters for collecting duct, clusters for endothelial cells. And then what we are doing is back mapping using this volumetric tissue exploration and analysis program. We are back mapping it onto the original image. And so this is what you see on the very right, where that truly overlays with the actual image of the fluorescence intensity that we get from the microscope. And why is this important is that we are able to take the actual microscopy image and we are able to transform it into an information that we can then use downstream for analysis of those images. So speaking of analysis, we can analyze each of the microscopy images separately. We can pull out that data, that fluorescence intensity, information about each of the markers, and then combine all that together and represent and analyze. Or what we have, our new approach is to use what's called combined analytical space. So rather than analyzing them each off separately and then combining at the end, we are essentially taking all the values, all the data, that information we are getting from each of the images, and combining them all together in an analytical space. Why does it have an advantage? Because it allows us to normalize the numbers and the data to allow us to scale when needed. So we are really truly able to have a more holistic approach to the data, and at the same time make sure that everything is standardized when we actually compare different images coming from different imaging sections, for instance, different imaging days using sometimes markers or antibodies, which could be changing too. So this definitely offers some analytical advantage. And so this is the model that we decided to use going forward. And here are examples of different tissues that we are utilizing. So you will see those very thin needle-like biopsies from patients with CKD. And we'll also have bigger tissue sections, and those are nephrectomies. Those can come from healthy patients, healthy donors, or also from some of the still diseased tissues. So we have a wide variety of tissue shapes and sizes coming from both patients with a kidney disease as well as healthy reference. So knowing all that, knowing that in our research we are utilizing cells coming from both the disease patients as well as healthy references, we're able to analyze those two big kind of cohorts of different tissues into that one combined space, and which resulted in this particular diagram that you see on the left. This is a landscape of all the distribution of all the clusters of the cells that you find within kidneys. And so you'll see, I hope I had to have a mouse here. Yes, I do. Perfect. So you will see right here, this region, that's the distal nephron. This region right here, that's the thick ascending limb. And those cells in both regions are actually very close together within the nephron. And so they tend to cluster together as well. Now here we have the proximal tubule. So those come from the region very close to the glomeruli. So it's right, this tubule right here. Now in the bottom, you will see the endothelium, the T cells, the fibroblasts, and immune cells. And again, those are clustering usually together very often next to the glom, because gloms are typically associated with a very high density of vasculature. So that's why we see these endothelial cells, which are closely associated with nephrons in general, just within the entire kidney structure, but gloms as well. So with that, we were able to name every of those clusters specifically as proximal tubule cells, glomerular cells. How did we do that? So other than relying on, of course, back mapping, we used the Violin plots, which you see right here on the right. And so with those plots, we looked at the fluorescence intensity and essentially how much protein was there in each particular cluster. Was it a cluster which was rich in podocalixin, like for instance, this one right here, which is a marker typical for glomeruli. And so aside from looking at the expression, the highest amount of expression or presence of a specific protein, we also looked at what protein is not there, because lack of presence of a certain protein in a particular cell is also informative and helps define that particular cell cluster. So I'd like to introduce couple proteins, couple markers that our lab has been interested in. And so we decided to add that into our codex panel. In addition to our structural, our immune, our injury markers, we added the following two. They are both cell surface protein, and they are both stem cell markers. So Thy1, also known as CD90, is a stem cell marker present on hematopoietic stem cells, mesenchymal stem cells, cancer stem cells. It's also found on neurons, endothelial cells, fibroblasts, and T cells. And this is actually where it was first discovered. So the role of CD90 is cell migration, it's cell adhesion, it's signaling, and of course, it's a nerve cell function. And then the other protein is PROM1. It's also known as CD33. And also, as I mentioned, it's a stem cell marker, also present on neurons and on endothelial cells. And its role is mostly in cell migration, proliferation, differentiation, signaling, and it's also responsible for the structure and function of cilia. So we were very interested in those two markers, and as I mentioned, we added them into our codex. And so we have recognized that we see those markers present specifically on proximal cells. And so as a reminder, those are the proximal tubule cells, are the cells right after the glomeruli. So we pulled all the clusters, all the cells, which were only the proximal tubule cells, and we decided to subcluster them again, just to see more granularity and see how much more deeper we can really dive into in defining each of those particular proximal tubule cell populations. So, of course, we again established or used the Valiant plots, and we were able to identify 10 distinctive populations or clusters of proximal tubule cells. Specifically and importantly too, we did identify, and you will see this on the bottom here, clusters which are positive for Thy1 and PROM1. So Thy1 has a specific, almost a separate cluster from the entire mix of other PT segments and classes. And then PROM1 is almost at the other end of that, where the Thy1 is found. And so you will see that by the increased expression here in red in those bottom panels right here. So as we talked about looking at the proteins, I mentioned earlier the spatial transcriptomic tool that we are utilizing as well. And so this is an example of it. We have taken two adjacent sections of the kidney tissue. And so this one on the very left right here, this is a kidney section which was analyzed using Codex platform. And then the one in the middle, you'll see the H&E staining kind of shining through the tissue here. And so this one was analyzed using or processed using spatial transcriptomics. And we're able to then co-register those two methodologies. So using protein and then using RNA, we're able to superimpose basically both images onto one another. And what we have discovered is that the Thy1 here in yellow on the Codex image is really corresponding to the yellow, the Thy1 yellow regions where they essentially are present within the Thy1 as the RNA. So on the far right, you'll see those dots. Those are those dots I mentioned earlier with the RNA expression. And so the more red the dot is, the higher the Thy1 expression. And so as you look at the yellow and the red, those really seem to correspond to one another. And so what that is truly confirming is, and supporting our finding, is that they are two complementary methods which are finding very similar results of where we see the regions of high Thy1 expression in the codex. We see the same regions using a different platform, in this case, spatial transcriptomics. So this is an example of back mapping, and we have the reference tissue, so the healthy tissue on the top, and then the diseased tissue on the bottom. On the far left-hand side, you will see the white nuclei represented by the DAPI staining, and you will see tubules marked in blue using the LRP2 marker, and that marker labels proximal tubule cells. In the middle panels, you'll see the Thy1 marker in purple, labeling several of the proximal tubule cells. And we also have a staining for PROM1 in green. Now, what's interesting is that in the reference tissue, you'll see predominantly this purple color, whereas in the disease, that purple color is less pronounced, but you will see a lot of the green staining that's indicative of that PROM1 presence. And so then when you look at the very far right side, you'll see that upon backmapping, we are able to create this overlay of just the nuclei, which are then further clustered down for downstream analysis. And those also really overlay and match of what we actually see just doing an actual staining for the protein in the tissue. So keeping that in mind, we have looked at combined, so all the tissues, the reference and the disease tissue, and we're able to distinguish those two separate clusters of Thy1 and Prom1. And we decided to map now the presence of those Thy1 and Prom1 markers onto a reference and a disease state. And we do notice that the amount of that protein present in the proximal tubule cells in a disease state has markedly decreased. And so this is visualized right here in this very faint blue part of the clustering. And so we took this a step further and we added more tissues. We added 49 different tissues from both HubMap and KPMP consortia. And this time, of course, we also have noticed the same or very similar trend in the presence of Thy1 protein being markedly decreased. But what's also important in this particular context is that in order to obtain this data and analyze this additional 49 different tissues, we have utilized an updated version of Codex, which is called PhenoCycler, and which has a lot of advantages as well, and a lot of differences too from the main Codex, one of them being that you no longer adhere tissue to a cover slip, you now use a microscope slide. It's also a little bit faster when it comes to imaging and processing. So essentially we are using two different, or still within the same principle, same idea, but kind of different modalities of Codex and PhenoCycler, but both yielding very similar results. And so these examples of images come from the PhenoCycler. So here on your very left, you'll see the reference tissue, and you'll see in blue the staining of the proximal tubule cells, which are very high in this Thy1 marker, Thy1 protein. Now in the red, those are the glomeruli. We have a little bit of green, but it's not very pronounced. Now, as we move towards the right side, we go through the AKI, so the acute kidney injury. And here you're able to see that we see a decrease in that Thy1 protein. And we start seeing a little bit more of this Prom1, kind of this transition towards that CKD state. And now as we look at the CKD state, this particular kidney biopsy, on the very right, you'll see a significant increase in this Prom1 staining in the green pattern, and a marked decrease in the Thy1 staining. So further, we also implemented trajectory analysis. And so in this case, what we are looking at is a starting point of using healthy cells, healthy proximal tubule cells for both reference and cohort and a disease cohort of cells. And so what we are able to appreciate here is that the trajectories of each of the reference and the disease are clearly different within, of course, those groups. And so that truly validates that those cells are in different cell states, essentially, those proximal tubules. We then took the trajectory, or we looked at the RNA data, and we applied similar trajectory analysis. And this truly confirmed our findings that those are really separate trajectories that are really going from the healthy to the injured and to the Thy1 marker. And so what's also interesting about this finding is that trajectory analysis has been most commonly done on RNA data, so single cell, single nuclear data. That's how it really became popularized. And so we took this method and were able to apply that to protein data, to essentially data which we collected from microscopy images, and had very complementary findings between those two. And so finally, as we move from the understanding of just specific cells and knowing the markers or the lack of the markers present in a particular cell, whether that's proximal tubule cell, whether that's a collecting duct cell, we then wanted to look at a little bit more of a broader picture, because it's important, not just a single individual state of a particular cell, but it's important who is nearby that cell. Is that a collecting duct that's injured and that's perhaps close to a lot of immune cells, or is it not? And that information is actually going to help us understand the progression from healthy to the kidney disease, as well as potentially elaborate on potential treatment options and find a way how we can really ameliorate or prevent potential injuries in the kidney. So understanding the neighborhoods is crucial. And so our approach is to take the cell-centric approach. So essentially, we are taking a nucleus, which is a fiduciary for the cell. And so then we apply a specific radius of interest. And then all the cells within that radius are being considered as one neighborhood. And then, of course, all that information, that data can be plotted in a very easy to visualize manner. So there are different types of plots which you can use to really represent those different neighborhoods. And so, for instance, the plot on your left, that shows a dimensionally reduced set of neighborhoods using the t-SNE plot. And so you'll see all those N1 and 2 and 3, those different neighborhoods and how they come together and who they are close to in terms of the entire layout of the kidney milieu, kidney environment. So here we have both the nephron cells, so those glomerular proximal tubule cells, collecting duct cells, but we also have endothelial cells, immune cells. And so now on the right is a little bit of a different way of representing the scattered plot. And so here we are looking at the main clusters of different cells and really looking at which cell type is the most abundant, which cell type represents the most of that particular cluster. And next, we also established this figure of the stacked bar plot, where each of the colors represent different type of cell. So for instance, we will have several of those bars for different types of PTs, for immune cells, for instance, endothelial cells, fibroblasts, and so on. And just to keep in mind that each of those particular bars, the way it's labeled is labeled based on the most abundant cell type within that particular cluster. And so once we do the odds ratio for the reference and disease, because of course we have that information, so we can pull that out from our data set. And so we can clearly determine that there are some specific cell types or neighborhoods which are specific to the disease state versus some other ones which are more common in the healthy or the reference state. So now, since our interest, as I mentioned in the beginning, was those two stem cell marker proteins, the Thy1 and the Prom1, we specifically looked at those. And so it appears that in N3, which is the neighborhood number 3, which contains high proportion of Prom1 with the Thy1 protein, Thy1 positive Prom1 PTs, that particular neighborhood also contains a higher proportion of proximal tubule cells, which are within the healthy boundary healthy state, and significantly less of the myeloid and immune cells, for instance. However, in the neighborhood 17, if you look at this dark purple color right here, that represents the proximal tubule class, which is heavily represented by the PROM1 marker, the PROM1 protein. And so in this case, we see a diminished representation of the different PT classes, as well as increased presence of the immune cells. So with that, it's important where you sit, just like at the Thanksgiving table. And so truly knowing the neighbors to a particular cell can truly get us closer to understanding how does a kidney progress from healthy to a disease state. It will get us closer to understanding the pathologies and then be able to through that understanding, to design ways of either preventing a specific kidney disease or potentially providing a treatment for that specific disease, because we will know the underlying processes and pathways that are involved by knowing all those neighbors and the cells that are involved in a particular play, especially if it relates to an injury area and the process of the differentiation and the fibrosis and scar tissue formation. So all in all, in this talk, we went from really the big picture. So looking at this three-dimensional tissue and being able to visualize the entire tissue section using different markers, different antibodies, where you can visualize different parts of that nephron with the endothelial cells and the immune cells, and really look at those correlations and the associations between the different structures within the kidney. We can then take that and look more specifically at a cell. And so then as we can look at that cell, we can then look at that cell state, and we can define whether that cell is dividing, whether that cell is a healthy cell, whether that cell is injured, but it's on the way to recovery, or whether that cell is injured, but it's actually going towards just a permanent injury state, and it's not going to be functional anymore. And so the thing that is also something to keep in mind is that we really are starting to understand that there are not just a few or several types of cell types within a kidney, like the proximal tubule cell or collecting duct cell or endothelial cell. In fact, with these findings, we are able to see that there is so many more cell types, and because those are expanded to include the cell states and the injury states. And so now we can have a proximal tubule cell that's perfectly healthy. We can have a proximal tubule cell that's about to divide, or that's undergoing an injury but regeneration at the same time. So, so that really is crucial to understanding the function of the organ and understanding the progression towards disease. So thank you.
Great. Thank you so much, Daria. And thank you. This was a phenomenal presentation, and Katy would love to get feedback and get questions. And thank you.
I was totally fascinated. You make some of the most beautiful data, and even going over to 3D slowly and steadily. So some of the questions I had were answered by Tarek, Daria. So I was thinking, well, you could have a coupled display, right? You could have your tissue data, imaging data on one side, and you could have the UMAP plot on the other side, and then you could select any part, right? Any cluster, even with a lasso. It doesn't have to be just a rectangle, you just have a lasso, and you get to see where it is on your tissue, right? And similarly, you could select a certain structure in your tissue, and you would see where it is in your UMAP plot. And this is absolutely feasible. And in fact, Felipe, who is going to present in the next hour, he has probably implemented many of these data visualizations because that's what he does, but also for networks, which I think is cool because ultimately it's networks of networks we're talking here about. So yeah, let's talk more. I think we have the team here in Bloomington to build this, and then you have the expertise to use it wisely. I had a different question. So there are stationary cells, and which are kind of stuck with their neighborhood, right? They can't move out. It's not like us that we can just leave and go to a different city, country, etc. But they are kind of where they are, and they have to make do with whatever they have surrounding them, be it senescent cells or aging cells or good cells. But then there are also transitory cells, and they are replenishing, they're coming through. And it's my understanding that immune cells might be of both types. And I wonder, how would you distinguish those two? Because it seems that they're different, right? And I wonder if there is a good way to distinguish those two types of cells.
Well, I can take a crack at this. Yeah, actually, this is a very important question. We're hoping that uh, particularly the immune cells, we think they are the messenger between the neighborhoods. And I think one of our goals in this work, particularly in HubMap and KPMP, is to really find the immune system based on what you said, Katy, based on the fact that which neighborhoods are they present in, because sometimes they could have the same marker, but they would be interacting with different players. So I think that gives them maybe a different function. That could be like a different. They're doing different things. So I think. As a first stab at this, we're trying to really look at the immune system as the link between these different neighbors. Now, of course, there could be other cells, other cells from the circulation or other de-differentiated cells that could also do that. But I think as a first stab of this, we really think that the immune cells could be particularly of interest. In particular, there's a lot of intravital data where people look at, for example, mice or rats under the microscope alive and look at the kidney, and we see that the macrophages move and T cells move, and I mean we all know that. But I think how they communicate, we do think that they actually communicate between different areas, and of course we don't know if they move also between what Daria showed, the cortex versus the medulla. I mean that's a very cool idea, is to track these cells eventually. One way is to think of doing a kind of more of also a trajectory analysis of these immune cells and see how they change and how they're interacting. That's something we're actually very interested in.
And so that's also where the neighborhoods can come really handy as a particular method of analysis, because even though you see a macrophage in a particular one region of the kidney, that doesn't mean that that macrophage is going to be identical to a macrophage that sits next to a glum, for instance, right? So that's where the neighborhoods are really, really useful.
Yeah, and we had a really interesting panel on cells and vasculature distance distributions earlier at 5 p.m. and lots of cool data across organs. And we are interested to see what cells are close to vasculature or to nervous cells or to other immune cells, which immune cells like to hang out next to other immune cells. I think this all becomes now possible with that rich and beautiful data that you just presented. Thank you for all the work on this. I know it's not easy. I wonder if there is a way for you to take kidney tissue and expose it to toxins. So obviously it would not be in a human being any longer. It would be probably from a organ transplants that can't be used. And then, however, introduce certain types of damage and then take samples before and after the intervention. Is this something you can do in your environment?
We're actually doing it in experimental model, like mouse and rats. I think that's and I think a lot of those we're starting to do these kind of instead of doing pseudo time, we're doing real time. Basically, we're injuring and looking at one day, 2 days, 5 days, 1 week, several stuff. And I think establishing an experimental timeline and see if the human timeline kind of tracks with that, then the experimental model will be a good surrogate to study kind of interventions in a way. I think establishing the human timeline is very difficult because we don't, I mean, we're humans, we eat stuff, we consume medication, we consume food, all of these affect a lot of things, right? But in a way, we can build some fairly reasonable assumptions that, if you encounter or if you have a certain disease, you have a certain kind of environments that are present, but if you are early in the disease, you have other environments that are present, and there's overlap between the two, and that overlap could be replicated in an experimental model. And I think these are some of the ideas. I think we need to prove that, but I think those are some of the things that are important, because many times we don't know where a patient is on the timeline of disease. And I think that's going to be important to really put the patients on a certain timeline, because if we want to do, like, for example, targeted therapy, we need to give the right medicine to the right patient at the right time. And that time is crucial part of that equation.
Yeah, and I hear you loud and clear that people, it might depend on how much water they had on that day, and what kind of stress they had at work, and what kind of emotional challenge they had, maybe also.
How many Zoom calls?
So yeah, right. But I actually enjoy that here. So this is a good time of my year. But it is interesting then to see in how far you could use printed organs, which might become available in the next 5 years, or transplanted organs, which you take out of the human body, but then kind of nourish, give it the
Yeah.
optimal environment to make them healthier, basically,
So,
before you implant them, right?
yeah,
So
yeah.
if you
So
could do
yeah.
that on those organs, that would
Yeah,
be
yeah, no,
giving a
that's
new
why there
material.
are a lot of groups that are using the organoids, where they're using actually, like, iPS cells, and then they're kind of trying to make, organoids that are very, at least in some aspect, they are similar. I mean, the kidney, there are a lot of beautiful kind of groups that are making these, and this is where you actually could do these types of intervention, and there are a lot of data that is produced, particularly for these toxicity, to figure out, or replicate, if you want. Do some interventions, do some perturbations in the organoids, that would really kind of give you a system model where you can test some of these associations that we see when we do these. And I think you're right on, Katy. I think that's the key thing why some of these models are important. Of course, we like in vivo stuff, but I think the organoid systems are very crucial also. They're very important to test these kind of toxicities, perturbation, and also some interventions, drug screening. What are the, there's a lot of, like, libraries now where you can kind of screen a lot of drugs on these things, where you can also see the different downstream kind of changes in the transcriptome and hopefully the proteome.
Yeah, and through CFAR, which is funding this event here partially, definitely, they have connected us to teams at the University of Toronto, which is just an hour away from Indianapolis, which actually are taking lungs that are meant for organ transplants, and they keep them in a warm environment, basically nurse them to be more healthy than they were when they got taken out of a human body. And they can keep them in that environment for quite a while. So I don't know if it's a week or 10 days, but it's a longer time. And so I think this makes it then possible, if it cannot be used for transplantation, to use them as a study project. And a lung is large. You could do a lot of experiments on that one lung. But of course, I don't know if that's also in existence already for kidney. Maybe that's just coming.
No, I think that there are some. For example, I do know that the University of Michigan group, Matthias Kressler, they do have a solution where they preserve the kidney in this cryostore solution, and that allows them to do, for example, single cell analysis at a later time point. They don't have to do it like immediately. So I think there's a lot of interest in preserving some of these tissues so that later you could do kind of some analysis. And I think you're right that that's going to be important, particularly if you want to, either grow these cells later or do something, whether transform them in a certain way. So I think those would be very interesting and eventually crucial to really validate some of these things that we're discovering, particularly in human tissue.
Yeah, so in ZenNet, which is the project that studies senescence, we have that set up for mouse and soon also human, where we take smaller tissue samples, like punch biopsies for skin. And then you can use 5 different ways to induce senescence and 5 different tissues of a human body or mouse body, and then look what it does before and after. And so then we would run Codex and RNA-seq and other assay types before the intervention and then twice after the intervention. So that, I think, is the kind of setup I would love to see for the atlasing effort. And then, of course, you can also compare across organisms. So you have mice and you have human. So the closer you can get to these kind of setups, and I know every single data probe is super expensive still. So if you do one before, two after, and then you do this across 5 organs and 5 interventions, so very, very quickly you have many, many dollars that are needed to do these kind of analysis. So it really takes these large scale common fund projects that NIH is now funding to get this to work, because much of the data that comes to teams just goes right into data production, and then we all benefit and get to see these beautiful images.
Absolutely. I don't think we can do anything at a scale that is going to be impactful alone. I think it has to be done as a part of a teamwork and, funded through kind of large efforts, just because we're seeing not just the technology expensive, the expertise and analysis is really not in one place. We have to really, combine efforts, but not just analysis, data visualization, make it digestible to in a way that is useful. I think those are all important points that we cannot do alone.
What kind of tools would you wish for if you would have all the resources on this planet? So what would be the most useful? Maybe you just need more money for data generation, but maybe there's something else that would be super valuable.
I think that not just, and I'll also let Daria weigh on this, but I think beyond data generation is important. Of course, there's but also, I mean, analysis and computational. I mean, moving data these days is becoming very difficult. Like if we had like a cloud like cloud servers available for everybody, where you could just take a volume that is terabytes and just render it and play with it and like do these interacting where you put it you segment the cells quickly, put it in a UMAP and starts to interact with it. Imagine how what an amazing advances we have. And yeah, and also I mean, I think visualization tools are needed. I mean, some of the things that you mentioned, because really we discover a lot when we are visualizing the data. A lot of the times we don't understand the data until we go through this entire pipeline. I don't know, Daria, what is your thoughts?
So the one thing I want to say is that we've been very fortunate to have some outstanding collaborators that we get to work with. So to bring that brain power and just really kind of brainstorm and bounce ideas off of each other, that's been crucial and that's been really amazing. But I do agree with Dr. Ashkar as well that just pushing the data from a place to place, really having a good infrastructure to store the data, to analyze it, that can sometimes be the bottleneck. And so we have great ideas, we have great ideas what to do in the future and how to analyze the data. But then it's really the storing and the actual analysis, which sometimes can run overnight or over 2 days, and you're just waiting for it, right? So so really that computing power is a big one.
One other thing, Katy, I think we really need to start training like it's like I think a lot of the times the training in informatics is separate from biology. I think these fields needs to be kind of really we need to create these tracks that train people into a computational kind of a computational physiology, computational medicine. I mean, this is crucial right now to generate the next generation of researchers.
Yeah, and this is a good time point for it, right? Because now we have IU Bloomington, IU Indianapolis, and there's a lot of interest from our
Yes,
leadership, definitely, to make
absolutely.
that work. So yeah, let's definitely talk more. And I hope you can stay for the next session. I think you will enjoy seeing what Philippe can do. And maybe next time you visit, we arrange a meeting with him also.